Hello, my name is Yoojung Kim, and this is my presentation on label-free microarray imaging by Oz Kumar's lab. DNA microarray is a versatile and effective tool that can determine mutations or changes in a DNA sequence, extent of gene expression by using RNA instead of DNA, and be able to quantify this data as well. Recently, this technology has been promising in clinical applications as well, such as determining if a patient has a certain mutated gene for a type of cancer. In that application, the scientist obtains the patient's DNA as well as a control DNA without the mutation in question, and then denatures and cuts the now single-stranded DNA into smaller chunks. A fluorescent moiety is attached to the small DNA chunks, and both samples are inserted onto a chip that is dotted with synthetic oligonucleotide targets that represent normal unmutated sequences on one side and mutated sequences on another. If the patient has a mutated gene, their DNA samples will not bind properly to the targets on the chip that represent normal sequences, but will instead bind to those labeled with mutated sequences. While this method is effective, it is difficult to quantify the extent of gene expression and relate fluorescence to the different binding energies that different mutated sequences have. Therefore, it is difficult to obtain repeatable results, and the reliability of this assay is questionable. The image on the left shows the result of two trials of the same gene transcript using conventional DNA microarray techniques with fluorescence by Dragici et al. The two graphs not only show that there are different results for the same transcript, but the variability is extremely large even within each trial. Oscamer's group had developed a technology published in a previous paper that could quantify and detect interactions of biomolecules in 2008. In this technology, as shown in image A, a laser is shined onto a silicon oxide top surface, and the reflected light is imaged on a camera. If there is a change in the height of the surface as a result of accumulation of biomolecules, the optical path will differ between the top of the surface and the lower silicon base. Once the image is taken by the camera, it is recorded as an intensity image over varying wavelength steps to form a reflectivity versus wavelength curve for each pixel on the recorded image. The result of this would be a reflectivity versus wavelength curve for each of the different optical paths. The data from these curves can be processed to show the thickness of deposited biomolecules per pixel. In the current paper, Oscamer's group demonstrated the efficacy and accuracy of the SRIB, or spectral reflectance imaging biosensor, by immobilizing target oligonucleotides onto the silicon oxide surface in a microarray, which would hybridize with specific nucleotide sequences that is presented to the array and lead to a varying thickness of deposited biomolecules. This figure shows the result of an assay done with our device. The before image in A shows the microarray dots before the assay was done. Image B shows the microarray after the assay. They were able to find that there was a significant increase in mass density on the dots after the assay compared to before the assay. Additionally, they were able to find that perfectly matched sequences show close to 100% efficiency in hybridization. The left image shows the kinetics of DNA denaturation based on the concentration of the ionic buffer used. The total drop in mass density is dependent on how much hybridization was present before the washing occurred. Stability of the hybridized duplexes can be determined by analyzing the slope or rate of mass density decrease. The image on the right shows the kinetics of the DNA denaturing during the wash with ionic buffer. This was done to determine the decay constants of differently matched duplexes based on their stability. The data from the previous assays were used to determine the final decay constants of different DNA duplexes with varying degrees of mismatch. This assay shows that a single mismatch is clearly distinguishable from a previous perfectly matched sequence when comparing those sequences with the same size as shown in B. In C, they showed that the size of the sequence mattered, as a shorter sequence tended to be less stable. In D, they confirmed that the stability of the perfectly matched sequences on their chip was the same as the stability of sequences before spotting. In conclusion, the Oskimor group used a technology they developed earlier that used analysis of optical paths reflected from a surface with deposited biomolecules instead of fluorescent measurements, and validated its performance. They showed that not only are they able to show mass density change after hybridization, hence allowing them to quantify the extent of hybridization, but they were also able to show that their device can distinguish single mutation sequences, which is important in clinical applications. Future works they might consider is to compare the mismatched sequences with different lengths as different length sequences have varying stabilities. Additionally, they should compare these mismatched sequences when there are more than one or two mutations, as mutations in actual patients are never as easy as only one problem. Therefore, they must also validate their device in a clinical study to show clinical relevance and prove reproducibility and low variability. Thank you.